The 11th ECR ICMRBS webinar. I'm sure almost everybody here knows how this works. So we're going to have 3 very quick talks from our speakers, followed by discussion in the breakout rooms of each of them. So I'm just going to pass straight on to our first speaker.
Okay, can you see my screen?
Yeah.
Okay, good. So hello, everyone. Thanks, Enrit and the rest of the organizers for inviting me. And today I will talk a bit about crowding and domain swapping. And I would like to start with an analogy I like a lot. So normally when you see my face in this picture, you would guess why this guy is smiling. You would know this by the context. So you know that this guy is surrounded by his favorite toy, the NMR magnet. That's why he's happy. So the same thing happens with proteins. Proteins act inside the cells, but the function of these proteins is highly dependent on its environment, also called kinase structure. And if you see these nice illustrations from David Gossel, you would say, okay, we know everything and we know all the colors, but in fact, all this, it's full of blacks and whites. So in order to understand that, we use nuclear magnetic resonance because we know that the nuclear spins are highly sensitive to the chemical environment. So basically, and just long story short, the crowding can be understood as a mixed contribution from 2 effects. One is excluded volume. Basically, 2 proteins cannot occupy the same space. That's why if you are in a crowded environment, you will favor compact conformations or preferential or weak interactions with the protein surface, either exclusion or inclusion in the surface. And it's dependent on the solvent accessible area differences. And those can be stabilizing or destabilizing. So crowding effects affect all kinds of features of proteins that affect protein function, either from protein folding or protein oligomerization or even protein dynamics. So the buffer you are in, it's important to understand the function. As the kinase structure, it's important to understand the proteins inside the cells. I just want to mention that on these different fields, there is plenty of excellent literature. But today we will navigate in a topic that perhaps is not much explored, that it's domain swapping, or as much as the rest. That it's basically a kind of protein oligomerization that requires a partial unfolding of part of the protein that exchange one domain with another to create an oligomer. So our system we use, it's just a model system. It's a mutant of protein L that goes through a partially unfolded intermediate to create a dimer. All these domain swapping events are highly interesting to understand many of the amyloid fibril formation phenomena that happen in several diseases. Importantly, domain swapping dimers exhibit extremely slow interconversion. And this oligomer stability can be understood in terms of excluded volume or preferential interactions, but also the protein shape. So the nice thing is that this system, you can separate monomer and dimer in the size exclusion, and you can quantify how much of the dimer or how much of the monomer you have. Also, this system gives a really nice HSQC spectra where you have 2 sets of signals, one corresponding to a monomer and one corresponding to the dimer. And in fact, the equilibrium is virtually the same as the one you would measure from size exclusion chromatography. So basically, you can start either from the monomer or the dimer and measure the peak volumes over time. So it's basically a real-time NMR experiment. And to a good approximation, you could assume that you have 2 monomers that form a dimer, and you can fit these peak volumes to the steady state kinetic equations to extract the kinetic rates. And then you can do a kinetic study on this and also a thermodynamic study. So basically we have a system like this. So we have 2 monomers that undergoes partial unfolding to create the intermediate, then an encounter complex to form the dimer, and the way back. Here, the orange bar indicates the degree of the different kinetic rates. And then we measure those in presence of different co-solutes of different nature, either sols of the Hofmeister series or also osmolytes that are organic molecules that cells synthesize either to stabilize the proteins or not, and also trehalose or sucrose that are sugars that are typically used in these studies. So if you measure the real-time NMR kinetic study, you would say, okay, you will reach an equilibrium. But for some, like sucrose, you would say, okay, but the equilibrium is not reached in those cases. What happens? So don't let the eye mislead you. In fact, the equilibrium is reached, but you just have to change the scale you are putting in the x-axis. So if you put a logarithmic scale, you will see how the equilibrium is reached in all the cases. But in the homeostatic time scale, meaning the time scale where function of protein happens in cells, that it's between half an hour or 35 hours, you see an apparent equilibrium for the different populations of monomer and dimer. So if now we plot how are the apparent populations after these 35 hours, that we call it the homeostatic time scale, you will see that in the dimer reaction, the protein does not sense the cosolutes. So you basically have the same equilibrium as in the absence of the cosolutes, but in the presence of the monomer, you have final apparent populations for. So what's happening here? Basically, in this case, because the difference in the area, surface accessible area are so small, those can explain this, but the same should happen for the monomer. So what is happening here? So as I was saying, for the dissociation reaction, where you go from dimer to equilibrium, the final populations are the same as in the standard conditions, so meaning in the absence of cosolutes. And this can explain by the small difference in surface accessible area. So you could assume that the activity coefficients for the monomer and dimer are close to 1. And from there, you can extract the activity coefficients for the transition state. And then you can have a rationale how the different cosolutes are acting. So I think we will have plenty of time to discuss about it in the breakout room. But then the question is, what's happening in the association reaction from monomer to equilibrium? So you will see that for some, the final monomer population is close to the one you would expect in the after of cosolutes, but in some others, you have much more monomer. So basically, the reaction is not flowing to create a dimer. So we measure which are the hydrodynamic radius for the different cosolutes, either from those experiments or checking at the literature numbers. And we see a nice linear correlation between the association rate and the hydrodynamic radius of those cosolutes, except for the denaturance ones, the vanadium chloride, thiocyanate, or urea. So basically, in the association of monomer in the presence of cosolutes, we have an exponential dependence on the cosolute concentration, and it depends on this proportionality factor that accounts for how fast the two intermediates encounter. And this proportionality factor is proportional to the relative size of the cosolutes and the protein, and also the shapes. Sorry. So from temperature-dependent studies, you can extract which is the energy barrier to the transition state. And we calculated that this is around 25 kilocalories per mole, indicating that, in fact, in this system, you have a large unfolded state that it's in between the monomer and the dimer in the reaction coordinate. So basically, cosolutes effects on the monomer affect the kinetic rates due to interactions with the expanded transition state. So basically, you have diffusion effects in there. So though for some of them, you are kinetically trapped. And the same was observed in molecular dynamic simulations. So basically, as the conclusions, so if we have this system in the ideal condition, in the presence of cosolutes that do not affect the unfolding, like urea, you would have a diffusion bottleneck that will not allow two intermediates to form the encounter complex as rapidly. And in contrast, in co-solutes like urea, you are facilitating the first step, then accelerating the association and the formation of the dimer. So basically, when you start the reaction on the dimer, you are under thermodynamic control. But when you are starting from the monomer, this thermodynamic control is highly affected by a kinetic trap. And with this, I would like to finish this short talk with the acknowledgements to Oscar, who was supervising all this project and from whom many ideas came out, and also the rest of the members in the group who helped on this, and also for the simulations to Gonzalo and the rest of collaborators. And thank you for your attention.
Thank you very much. That was great. So let's pass straight on to our second speaker.
Yeah. Can you see my screen?
Yep.
Cool. Okay, so yeah, I'd like to start my talk by thanking ICMRBS for giving me this opportunity. I'll be talking about the contribution of electrostatics to hydrogen exchange in intrinsically disordered proteins, specifically human alpha-synuclein. I did this work as a postdoc in Aarhus University in Denmark in Franz Molter's group. So coming right to it. So hydrogen exchange, as we all know, is a very complicated process where the kinetics depends on the protein dynamics as well as on the intrinsic rate of exchange. But when there's a structure formation, the amide hydrogen exchange rates, they reduce relative to when the protein is in its solvent exposed reference state. And this reduction can be expressed as a protection factor. So your protection factor is intrinsic exchange rate divided by the observed exchange rate. And so the exchange kinetics depend a lot on the protein sequence. Molter and Englander showed the effect of side chains on the intrinsic exchange rate. And they did so by constructing libraries of tripeptides and measured the expected hydrogen exchange rates, and accordingly they built a model. Now, this model is quite accurate for unstructured polypeptides where the observed rate of exchange is approximately equal to the intrinsic rate of exchange. But for longer proteins, this model is only an approximation. But in this talk, we're going to talk about the influence of electrostatics on hydrogen exchange and give a quantitative description for the unfolded state. Now, there are a number of methods in NMR for detecting hydrogen exchange. In this work, we will be using the deCORE approach. The deCORE approach was introduced by Skrinnekov and Ernst in 1999. And they showed that exchange events with solvents lead to decorrelation of the involved spin operators. Ketub and Pilupisi in 2007 extended this method to detecting the decorrelation of the antiphase operators. And we're going to call this approach the Paris deCORE approach. In this method, CPMG spin echo trains are inserted on the nitrogen channels, while keeping the total CPMG time constant. So each time the total time is constant, we're basically only varying the number of pi pulses. And this helps us generate admixtures of in-phase and anti-phase operators. Now, every time there's an exchange event, the signal from the anti-phase operator gets decorrelated. There's a signal loss every time there's an exchange event. And this signal loss reports directly to our exchange kinetics. So every time there's an exchange event, the antiphase operator gets decorrelated and destroyed. And as the method does not rely on the detection of amide proton, we're able to detect fast exchanging residues at a very high pH. In addition, we also do a reference experiment where there's a Waltz decoupling on the proton channel, which helps us generate a spectrum that contains no exchange losses. If we compare the intensity from this spectrum to the intensities that were determined previously without the Waltz decoupling, we are able to get our exchange rates The methodology, the pulse sequence, and the code to do so is available in this manual that we wrote last year. Yeah. So in conclusion, our first conclusion is that yes, we have a method that helps us detect fast exchange rates. We've already used it in HackaCon and CON modules, and it's quite suitable for folded and unfolded proteins. But now we'd like to go further and study the hydrogen exchange rates on alpha-C nuclei. It's a very interesting protein with 140 amino acids. It's intrinsically disordered with 3 very interesting regions. You have the N-terminus, which is slightly positively charged, the NAC region, which is devoid of any charges except for these 2, and the C-terminus, which is highly negatively charged. And so we used our Paris-T-core approach on a 2D con pulse sequence, and we measured hydrogen exchange rates at pH 9. We varied the number of pi pulses on the nitrogen channel and kept the total CPMG time constant at 20 milliseconds. And we see a beautiful trend like this and a plunge at the C-terminus. Now, we can, as discussed previously, we can go to protection factors using K intrinsic divided by K observed, where K intrinsic is, like I said, from the Englander models, and you can get it at the Sphere server. The K observed is what we got from our 2D con experiment, right? And the protection factor shows this variation at the C-terminus. Now, protection factor of greater than 1 indicates a structure formation. But is there really a structure formation at the carbon terminus? Now, people have studied this before. Alexandrescu and co-workers had added 300 millimolar of NaCl to their sample, and when they did this, they saw that this enhances exchange rates and erases the difference in rates between the N and C termini. Okazaki et al. Acknowledged something similar, and they said it could be because of the charge distribution and the long-range interactions between the N and C terminus. So although we know that our intrinsic exchange rate model gives some inaccuracies, but these results have been well reproduced in previous studies. And still there is an explanation lacking for the structure formation of alpha-C nuclei, as well as impeded hydrogen exchange for the C-terminus. And so we turn to NMR chemical shift temperature coefficients, because impeded exchange rates due to hydrogen bond formation is accompanied by reduced temperature coefficients. And we can calculate temperature coefficients from the BMRB entry for alpha-C nuclei, as we did here for these 5 temperatures. A dashed line at -4.5 shows the approximate border found to correspond to hydrogen bond formation folded proteins. But for most part, we see a fluctuation in this region around -6 to -7, which is typical for unstructured regions. And so we can say there is absence of hydrogen bond formation, or we can also say that there is maybe some weak interaction between the N and C terminus, but that's about it. Now, to dig further, we went into paramagnetic relaxation enhancements, PRE experiments. And to give a brief introduction, PRE experiments are when there's a dipole interaction between the unpaired electron of a paramagnetic ion and with the nucleus of interest. And with this interaction, it increases the relaxation rate of the nucleus, which causes a broadening of resonance of the nuclei in the proximity. So we run 2 experiments, 15 NHSQC of alpha-C nuclei with a neutral gadolinium complex and a negatively charged gadolinium complex. And we want to compare the intensities between these 2 experiments. And when we do that, we saw this interesting trend again at the C-terminus. The negatively charged complex caused less line broadening at the carbon terminus, which means that the negatively charged complex does not approach the C-terminus to the same extent as a neutral complex. And this is in accordance with the electrostatic repulsion. Now, another way to quantitatively study the ion depletion at the protein backbone is to use the Debye-Huckel approximation for a Gaussian polyelectrolyte protein chain. So we compute the electrostatic potential using the hybrid mean field approach for each position I summed over all other positions j at pH 9. And we get a trend like this again, which is in line with what we got from our PRE experiments. And this helps us conclude something important. So at above pH 5, we know that it's the hydroxide ions that catalyze our exchange kinetics. And so a low concentration of hydroxide ions in the vicinity of polypeptide chain would result in a reduction of the exchange kinetics that we observe. So to answer the question, could alpha-synuclein's acidic tail be reducing the hydroxide ion concentration? We can go one step further with this, and we can calculate expected protection factors from electrochemical potential. So we all know this equation. At equilibrium, the electrostatic interaction energy acts to lower the local hydroxide concentration. So this computed potential can be used to estimate the change in concentration of hydroxide ions at the protein backbone and therewith predict protection factors, because our protection factor is proportional to the fold change in hydroxide ion concentration. Our protection factor is proportional to what we computed as our potential before. And so we computed this before, and we computed our expected protection factor here, and we see that the trends match. The trend seems in line with what we got previously from, as well as from our PRE experiments. And from the electrochemical potential, a 4-fold reduction in the hydroxide concentration near the protein backbone is predicted. And all of these is in conclusion with, is in line with also what we got from our NMR spectroscopy experiments. And so in conclusion, although electrostatic interactions between the N and C termini exist, they are weak and insufficient to protect amides from exchanging with the solvent. Negative ions do not come as close to the acidic polypeptide tail as the neutral ones, explaining why amide protons appear protected at the C terminus. And the reduced concentration of hydroxide ions around the acidic tail is a manifestation of the electrochemical potential. And with this, I would like to thank you for your attention. Yeah.
Thanks very much indeed. Okay, so we're going to pass on to our final speaker before the breakout rooms.
Hi, I'm Angus, a postdoc in the BACS group at the NIH. Thanks to the organizers for inviting me here. Today I'm going to describe some of our research efforts to characterize the main protease of COVID-19. Now, in the structural biology era, where EM is king, X-ray crystallography is queen, and NMR serves as the court wizard, how do we continue to provide useful insights to support the other techniques in our discipline? Current efforts to tackle virus prevent penetration into cells, either by administration of antibodies to prevent cell adhesion or by training the host immune system to recognize viral antigens. This new approach using RNA is potentially quite exciting, given the ease of adapting this drug development process to new spike protein epitopes as they arise. However, while prevention is almost always better than a cure, there's always going to be cases where pharmacology is needed. Now, after entry into the cell, two proteases process the two viral polyproteins. One, papain-like protease, cleaves at about 5 sites, and a chymotrypsin-like or main protease cleaves about a dozen sites. The main protease is the focus of this current study, as its low mutation rate and central role in the replication cycle of the virus presents an attractive drug target. Now, as of a few weeks ago, there were over 160 PDB structures of MPRO with or without ligand, with more structural diversity between structures than occurred upon ligand binding. This presents an attractive model for docking-based drug design. However, it raises the importance of docking into the correct structure. We wanted to use NMR to validate these structures, secondary structers, and potentially identify any minor or excited states that could be exploited. Now, the main protease is a 68 kilodalton homodimer with a correlation time of about 27 nanoseconds at 298 Kelvin. Now, this mandated pituitration for assignment and structural dynamic investigation. And one of the issues that took a long time to resolve was back exchange of buried amide resonances in the core of this dimer, highlighted in blue on the slide. The solution was to partially unfold and refold the initial fusion construct with an N-terminal solubility tag, because, at least in my hands, fully unfolding the protein led to almost total sample loss. Now, the enzyme mechanism involves a catalytic dyad of H41 and C145, and well, at least I struggle to effectively express and stabilize the wild-type variant of MPRO, the main protease. However, the C145A variant was stable for weeks to months. So all of the spectra presented here are recorded on that variant. Now, even at 900 megahertz shown previously, there's still substantial overlap in the TROSY spectra. The contours cut pretty high here. At the time of inception, there were dozens of high-quality PDB structures available. This time last summer. So assignment by NOE distance restraint was attempted. We implemented 2 4D experiments. The first was a TROSY experiment, where for a given peak, say here, that I now know is L232, we can take a cross-section at F3 and F4 frequencies in the reference plane. The resulting F1, F2 nosy plane displays cross peaks that correspond to residues within NOE distance, about 6 or 7 angstroms. Secondly, was a 4D nosy TROSY experiment, where 2 different nosy mixing periods, 50 milliseconds and 300 milliseconds, were used to determine the distance of F3, F4 cross peaks from each other in the corresponding solution structure. The displayed cross peaks arise from a 50 millisecond nosy mixing period, while the diagonal reports on the 300 millisecond mixing. Now, a quick note on implementation and feasibility. High quality reconstructions were readily achievable, using SMILE in minutes to hours. We observed that about a day and a half was optimal for either the double nosy TROSY or the TROSY nosy TROSY experiments, indicating that they could be a useful and informative inclusion to a standard backbone assignment repertoire, if not de novo assignments themselves. On the topic of resolving overlap peaks and addressing the structure validation mission statement that I described previously, we implemented an improvement on the standard 2D RTC experiment for residual dipolar coupling, RDC, acquisition. The RTC experiment comprises 2 spectra, a reference spectrum and an attenuated spectrum. The RDC is derived by comparison of peak intensity values in these 2 spectra. Notably, by introducing an additional carbonyl frequency dimension, turning your RTC peak picking from a crowded 2D to a much sparser 3D, you increase the RDC coverage of your protein. Here we use the 3D RTC RDC peaks supplemented with 2D RTC RDC peaks, where exchange broadening resulted in weak but resolved TROSY peaks. RDCs determined by 2D or 3D methods perfectly agreed within experimental error, with only about a 2-fold loss in sensitivity for the 3D experiment. Now, as I said, SMILE was used for the reconstruction, but the simultaneous reconstruction of both reference and attenuated spectra acquired using NUS for this TROSY HNCO RTC experiment to improve reconstruction fidelity. That was a lot of acronyms. Using these RDCs, we could perform standard SVD fit of RDC value to the whole population of high resolution crystal structures available. However, we see much higher Q factors than we expected with a relatively broad distribution. This is much higher than an expected 20% of good solution structures like ubiquitin. Further, the very poor correlation of the median predicted RDC against experimental RDC confirms some substantive rearrangement of the structure is needed to fit our experimental RDCs. Furthermore, if we plot the range of RDCs predicted against experimental RDC for an example region, we can see that there are entire stretches of the backbone with orientations not sampled by the PDB structure ensemble. If we plot these RDCs that disagree on the dimeric structure, they're distributed in clusters across almost the entire protein complex. However, if we fit the two domains separately, we get much better results. If we fit an N-terminal fragment in red and a C-terminal fragment in blue, we get much better agreement with Q factors on the order of 15%. This is a strong indication that not only is there some domain rearrangement, but also that there's some rearrangement within those two domains. While this isn't a therapy session, further complications of the assignment arose in the form of two separate minor species that were both present and prevalent. The first minor species is present for about 30 residues, colored in magenta here, and localized around P184, which abuts the P4 binding pocket of the active site cleft. Secondly, work in progress. Several residues indicated in green show a minor state with a different secondary structure, well, a different secondary shift, sorry, that additionally exhibits exchange broadening. This combined with the adjacent 40s loop, colored in blue, reportedly dynamic in MD simulations, indicates another consideration for drug designers. Now, while all is good and well, to assign it, we need to validate our assignments and to test that what we expect to move on substrate binding did in fact move, especially given the many possibilities for assignment error described previously. We added a substrate sequence analog, VDYLQ, which was expected to bind at the recognition site indicated on the slide. The titration was followed by TROSY at 900 megahertz in PBS over a range of substrate concentrations from 0 to 3 millimolar, and it was determined that the KD for this binding interaction was about 100 micromolar by Water-Logsy. We see that, as expected, chemical shift perturbations largely occur in the active site of the protein. Notably, perturbations were also seen at the NNC termini of the protein. Now, this was due to the nature of the homodimer, where specifically the N-terminus directly interfaces with the active site. Our native N-terminus reflects this, while the uncleaved fusion construct is unable to even dimerize. A final note to leave on is, are there potentially any roles for the additional states that we've seen? Because at least the major and minor states around P184 are in slow exchange, we can investigate these two species simultaneously. For example, here binding of the substrate peptide analog, both states bind with similar affinity indicated by the fast exchange. And even when bound to ligand, they don't collapse into a single state. And with that, I'd like to give a big thanks to Jimfer and Dusty for keeping the magnets running in these challenging times, particularly to Jimfer for help with SMILE and experiment optimization, to Yang and Charles for help with structure investigations and queries, Charles particularly for structure fragment screening, to Joseph and the rest of the BACS group for helpful discussions, and to Ad for supervising the project and keeping me on the straight and narrow. Thanks for listening.
Okay, thanks, Gus. That was great.